and to my dear students. We are thrilled to host our 5th session of Sci-Tech Spins Lecture Series, an initiative of the Office of Academic Outreach at IIT Delhi, which aims at enriching the educational experience of high school students. It is my honor to welcome you all today for Sci-Tech Spins Lecture. We have kept STS on 17th September as it is celebrated worldwide as International Microorganism Day. The day honors the legacy of Anton von Lucke, the father of microbiology, who first discovered the hidden world of microbes. Just as microorganisms may be small but powerful, today's lecture also aims to open new doors of knowledge and inspire big ideas. By holding this lecture on International Microorganism Day, we wish to emphasize the importance of scientific exploration, discovery, and dialogue. Just as microorganisms are the foundation of life, so too is the science the foundation of progress. A guiding star with knowledge deep, she sows the thoughts that we shall keep. Claps and smiles light up the air. Dear Professor, we welcome you here. It brings me immense joy to introduce you all to Professor Preeti Srivastava, our eminent speaker of the day from the Department of Biochemical Engineering and Biotechnology at IIT Delhi. She obtained her PhD degree from Department of Biomedical Engineering and Biotechnology at IIT Delhi in the area of bacterial genetics and molecular biology. She did her postdoctoral research work in the area of chromosome dynamics in Vipro Gloria from Laboratory of Biochemistry and Molecular Biology, National Cancer Institute, National Institute of Health, Bethesda, Maryland, USA. Her present research interest is in the bacterial genetics and environmental biotechnology. She has published several papers in internationally reputed journals and proceedings of national and international conferences. She has been granted 4 patents and has filed 4 Indian patent applications. She is the recipient of the prestigious Innovative Young Biotechnologist Award in 2013 and Tata Innovation Fellowship in 2024, both from the Department of Biotechnology, Ministry of Science and Technology, Government of India. She is also the recipient of Teaching Excellence Award from IIT Delhi. With claps of joy and hearts so keen, we welcome the voice that must be seen. Dear Professor, the dice is yours to share your wisdom and open new doors. Now I hand over the mic to Professor Preeti Srivastava for her valuable words. Ma'am, please.
Hello. Good morning to all of you. Am I audible in the last row? Yes, okay. So thank you so much to the Academic Outreach Office for giving me this opportunity on the day, which is a very special day. It is International Microorganisms Day. And so the title of the talk is Microorganisms, the Secret Superheroes of Our Planet. And before I begin the talk, I will give you the outline of the talk. So we will start with the introduction. Like, why are we celebrating International Microorganism Day? Then I will be talking about what are microorganisms and why are they called superheroes. What do we do at our department, which is the Department of Biochemical Engineering and Biotechnology. And then to make today's day so special, we have planned a few activities. One of the activity will be demonstration, which is view your microbes, where we will be using our own in-house microscope, which is manufactured by a startup from our department. And then this will be followed by do it yourself activity. So where every student, each one of you will be carrying out experiments, small experiments, and I will be telling you the basic principles and the basis of those experiments. This will be followed by announcement of prizes. So we conducted as part of International Microorganism Day, we organized 2 hackathons for school students as well as for college students. So we will be announcing the winners of that, and finally you will have a feedback form. So to begin the talk, how many of you know that what is so special today? So today actually is 17th September and it is celebrated as International Microorganisms Day because there is a story behind it. So there was a Dutch man, Antony van Leeuwenhoek. How many of you have heard this name? Okay, very good. So Antony van Leeuwenhoek was actually born to a businessman family. So his mother was a house person and his father used to make baskets. Antony van Leeuwenhoek actually, he had a shop and the shop was actually for manufacturing fabrics. Imagine, he was not trained as an educator. He was not having any higher degree. But then, during this time, he developed an interest of making lenses. You know what are lenses? Lenses, right? So he made a huge number of lenses, 419 lenses, right? And then he made about 250 microscopes. So, but then what is so special for 17th? So on 17th, Antony van Leeuwenhoek actually communicated a manuscript in Royal Society of Chemistry and published the paper where he demonstrated the unicellular microorganisms with the help of a microscope. This was his own microscope, right? So that is why 17th September is the date to remember, because on this day, for the first time, Antony van Leeuwenhoek discovered unicellular microorganisms and he called them as animalcules. Okay? Now, what are microorganisms? So microorganisms, as the name itself says, the organisms which can be observed under microscope, which are not visible to naked eye. So when we talk about the classification, we have viruses, bacteria, fungi, algae, and protozoa. Starting with viruses. Viruses are actually non-living, but when they enter a host, they start replicating and then they grow. So viruses, here I would like to mention that viruses which infect bacteria are called as bacteriophages, right? So viruses which infect bacteria are called as bacteriophages, and it is these viruses which are very, very useful, which are used a lot in genetics, which are used a lot in phage therapy. Now what is phage therapy? I will be telling you as we go on. The next category of microorganisms are the most interesting ones. These are called as bacteria. And when we talk about bacteria, bacteria are again, they come in various shapes. They can be rod shaped, they can be cocoid shaped, they can be spiral, right? They can be comma shaped. For example, Vibrio is comma shaped, Coccus is cocoid shaped, and then they are discriminated based upon gram staining. This is based upon the amount of peptidoglycan which is present in the cell membrane, and you will be doing this activity today. I will be discussing that. The third set of microorganisms are fungi, and in fungi mostly the most useful ones are yeast and molds. Mushrooms are also grouped under this category, but then they are not microscopic in size. We have algae, the next category. So blue-green algae are used a lot in studies. And finally protozoa, which includes Euglena, Paramecium, etc. So this was about microorganisms, but then what about microorganisms in our body and microorganisms surrounding us in the environment? So let us first talk about microorganisms in our body. Where do we have microorganisms? So microorganisms are present in our nose, they are present in mouth and throat, they are present in stomach, present in gut, skin, as well as vagina. And when it was calculated, it was found that the amount of microorganisms, right, the amount of microorganisms that a body has, you cannot imagine, it really outnumbers the total number of somatic cells in a human being, right? So I'll show you that. And these microorganisms, they contribute to digestion, they produce vitamin K, they promote immune system development, and they toxify harmful chemicals. We will be discussing this in more detail in a while. But what I have shown over here is that 10 microorganisms per every cell. Particularly, so although I showed you that microorganisms are present in multiple parts like nose, throat, stomach, but then the number of microorganisms in the gut, this outnumbers all other places. So in the gut actually the total biomass of microorganisms which are present in the gut is about 1.5 kg, and this is roughly the size of the liver. So you can imagine that the size of the liver is almost about the size of the total biomass, right, of the microorganisms. And the other important point to note over here is that there are 1,000 different types of strains, 1,000 different strains, although 160 of these are conserved in all. So that means all of us have about 160 strains which are common. But then the rest are all different, and this is how this governs the digestion, this governs the mood, this governs a lot of more things which I will be talking about in the next few slides. So this is a slide which shows the difference between microbiota and the gut microbiome. So remember I was talking about the biomass of the gut microbiota. So what is microbiota and what is microbiome? So microbiota is the community of microorganisms themselves, and the microbiome is the collective genomes of the microorganisms in a particular environment. So over here if you see, the human body has 10 trillion cells, but the gut microbiota has 100 trillion cells, right? So it's a lot more. Now, if you see human genome, it has approximately 23,000 genes, but the gut microbiome has 3.3 million genes. So it's a lot, right? So how is it helping us? Is it really helping us? Why are these microorganisms there in our body? Are they really controlling us? Are they really driving us? So these are some of the very interesting questions which people are trying to now understand. So starting with the gut, so what you see at the bottom, so over here, this is actually the intestinal epithelium, and over here is the indigenous microbiota, and what is shown is that this indigenous microbiota, this actually makes a film on the intestine. It makes a film on the intestine, and it is this film which prevents the invasion of microorganisms in the intestine. So for example, if there is a pathogen which invades the intestine, then it is this intestinal lining which protects the pathogens from invasion, right? And then this also produces small chain fatty acids. Now about the gut microbiota. So for that, I would like to share with you a superhero origin story, and I'll tell you why I call it as a superhero origin story. Because in 1917, during World War I, it was found that many soldiers were infected with Shigella, which causes actually a dysentery, right? But somehow, interestingly, one of the soldiers survived. Now the soldier who survived, it was found by a German scientist, and his name was Alfred Neisel. This is the name of the German scientist, Alfred Neisel. So Alfred Neisel realized that the reason behind the person who was not infected with Shigella, right, is something which is in the stool itself, which is in the fecal matter itself, and then he found that there was a bacterium which was E. coli, and he named that bacterium as E. coli Neisel, E. coli Neisel. And E. coli Neisel now also is used as a probiotic, and this is available commercially also. It is sold in the name of Mutaflor, and you see that the active substance is E. coli strain Neisel, right? So E. coli Neisel is actually a probiotic, right? It is not only helping in digestion, but it is preventing the pathogens from invading the intestine, right? So that is why these microorganisms are called as probiotics, right? So a term that is used conventionally is probiotics. Now, but then you see there are some terms which are used a lot in our day-to-day life. Probiotics, prebiotics, synbiotics, postbiotics. So what are these? So probiotics, as the name itself says, they are live bacteria and yeast, and they are commercially available in the form of Darolac that you buy whenever you have stomach upset. You buy Darolac, Visilac. So all of these, they contain probiotics. These probiotics actually contain live strains of, for example, Lactobacillus acidophilus, for example, Bifidobacterium, for example, certain yeasts are also probiotics. Now what are prebiotics? So prebiotics are actually the compounds, the substrates which actually help in promoting the growth of probiotics, right? So prebiotics can be, for example, if we are eating fiber-rich food, sprouts, beans, so some of those vegetables or some of those foods, when they are digested, they release certain compounds which can actually improvise the growth of probiotics. Okay, so probiotics are the live bacterial or yeast strains, and prebiotics are the ones which are promoting the growth of probiotics. Now what are synbiotics? Synbiotics are actually a combination of probiotics and prebiotics, right? What are postbiotics? Postbiotics are the ones which are actually dead. They are neither living cells, they are not substances which promote the growth of living cells. They are dead. For example, some short chain fatty acids, for example, heat-killed Akkermansia mucinophila is also used as a postbiotic. Now there is another term. There is another term, and that is psychobiotics. So what are psychobiotics? Anybody knows what are psychobiotics? Any guesses? So psychobiotics are actually probiotics, right? They are actually probiotics, but then they are the ones which affect your mood. So for example, some people are happy, some people are unhappy. You have different emotions. So have you ever thought of that it is the gut microflora which can govern this, which can actually control this? How is it doing? So these microorganisms, so-called psychobiotics, right? They actually affect mood, emotions, and your overall behavior. And how do they do this? They produce certain compounds. One of this compound is serotonin. What is a serotonin? Serotonin is a neurotransmitter. So neurotransmitters are actually chemical compounds, right, which can sense the signal and convey it to the brain. They also produce certain short chain fatty acids. So these are transmitted with the help of what is called as gut-brain axis. So gut-brain axis, and this gut-brain axis is controlled by what is called as a vagus nerve. Vagus nerve is the 10th cranial nerve, and this is the longest cranial nerve extending from the brain to the heart, lungs, and finally the gastrointestinal tract. And it is this nerve, vagus nerve, which senses the signals from the gut in the form of chemical messengers, and these chemical messengers are actually neurotransmitters. The word neurotransmitters because they are transmitting the information to the neuron, and thereby, so this is the figure which shows that these are the compounds. GABA is gamma aminobutyric acid, then serotonin, dopamine, how the signal goes from gut to via vagus nerve and how it controls your mood. Finally, gut microbiota has also been associated with longevity. So So for example, the neurotransmitters that I was talking about are acetylcholine, GABA, serotonin and dopamine, and they are produced by these strains like Bacillus, Serratia, Candida, Streptococcus and so on. All of these are bacterial strains. So that is another interesting thing I have to tell you, that in our human body, bacteria are predominantly present. So that is why you see all the names are mostly bacterial strains. For longevity, it was found that some bacterial strains like Odoribacter, Oxalibacter, Cristensenella, etc., they were found in the gut of centenarians. So these were the people who were living for more than 100 years. And then finally, the point is, I was talking about the microorganisms are controlling our mood, right? But then how it is the food which is affecting your mood. So the food is affecting your mood. How? Because it is the food which we are eating is actually allowing the growth, it's promoting the growth of certain probiotics, right? So for example, if we are eating a lot of food which is rich in, for example, fats, for example, you keep on having cheese, pizza, etc., or if you are having too much sugary drinks, then it will not promote the growth of the good bacteria. For the growth of the good bacteria, we need fiber-rich food, fruits, etc., and therefore it is the food which promotes the growth of those probiotics which actually serve as psychobiotics, and it is these psychobiotics which are responsible for a healthy brain. So this was about so far what we discussed was the microorganisms in our body, right? We discussed what is there in the gut and how the microorganisms in the gut are actually affecting the health and they are actually affecting the human brain. But now let us see the beneficial microorganisms in the environment, right? So when we talk about environment, so microbes actually run the world. You will be surprised to know that a large proportion of oxygen is coming from algae, right? We also have viruses, and as I was talking about viruses, whenever we name the word viruses, you would think about coronavirus, you will think about monkeypox virus, Ebola virus, right? All dangerous viruses. But have you ever given a thought that these viruses can also be beneficial to human beings? The answer is yes, these viruses can be beneficial for human beings and also for the environment, because as I told you, these viruses, they can infect bacterial cells, and when they infect bacterial cells, they can actually kill the bacteria. Such viruses are called as bacteriophages, right? Bacteriophages are the viruses which infect certain bacteria and then they can lyse the bacteria. So imagine if it is a pathogenic bacteria. If it is a pathogenic bacteria, then the bacteriophage will infect the pathogen and will lyse the pathogen. So then you know you already have a substitute for antibiotic. Instead of a chemical antibiotic, we can do what is called as phage therapy, because the phage will actually infect the bacteria and it will kill the pathogen. So that is why all viruses are not dangerous, all viruses are not bad, but then they can infect the bacteria. Yes, ask me if you have a question. Anybody has any questions can ask me in between as well, just raise your hand. Okay, so then about viruses, so this is how viruses look like, some of the viruses. Finally, bacteria. So bacteria also help in the environment. For example, they help in the fixation of nitrogen, they help in the increasing the fertility of the soil, they help in the growth of plants, they promote the growth of plants. So thereby they help in they act as biofertilizers, and in fact our outreach associate dean works in the same area. They also help in bioremediation, because they can help in degradation of certain pollutants, certain xenobiotics. They are also used for production of high value compounds. For example, certain therapeutics are being produced from bacteria. They are used for preparation of curd, cheese, etc. When we talk about fungi, they are also used. For example, in bread, the yeast is used. In wine, ethanol is used. They are also they produce mucoprotein. They also produce anti-cancer, antiviral, antibacterial compounds. Penicillin is one of the most notable discoveries of the application of the fungi, where Sir Alexander Fleming was awarded the Nobel for that. Finally, we talk about protozoa. So in protozoa, we have multiple species, amoeba, paramecium, euglena, etc. and they are detrivorous and they help in mineralization. And finally, algae. They also have multiple applications. Algae, as most of you are aware of, like spirulina is like a food. It is like a whole food. If you have a tablet of spirulina, it covers the entire nutrients, vitamins, minerals, etc. They are also used in biofuel production, carbon sequestration, pollution remediation, etc. So far we have discussed microorganisms in our body and microorganisms surrounding the environment, the beneficial microorganisms. But is that enough? So can we meet all our needs using the native microorganisms? So the answer is yes and no, because at times we want to overproduce. For example, if we are producing, for example, certain therapeutic, let's say insulin or let's say erythropoietin. So these are therapeutics, right? These are used as medicines, and can we overproduce these compounds? So for that we need to do what is called as engineering. So which is genetic engineering, and genetic engineering involves multiple plasmids, multiple enzymes. I will be talking about that in a while. So engineering microorganisms has been reported for viruses, bacteria, yeast, trypanosoma, and as well as algae. So this is a typical diagram which shows how we can engineer bacteria for biotechnological applications. So bacteria, so I am taking bacteria as an example, but along the same lines we can engineer other organisms as well. So when we talk about engineering bacteria, so bacterial genome actually consists of a chromosomal DNA, and it has an extra chromosomal DNA. So I hope the students are aware of what is a chromosome. Raise your hands. This side, nobody knows what is a chromosome. This side, okay. So then, so bacteria have chromosomal DNA, but then they have something which is extra chromosomal, right? And this extra chromosomal DNA is actually called plasmids. These plasmids are used as vectors. Why the term vector is used? Vectors because they are carriers of genes. They are carriers of genes, so that means the desired gene can be cloned in these plasmids, and then this plasmid can be transferred from one strain to another strain. We also use what are called as restriction enzymes. So restriction enzymes are actually biological scissors because they can be used for cutting the DNA. We use DNA ligases. DNA ligases are actually the glue. Why they are called as glue? Because using ligases, we can cut the DNA which was cut can be joined with the vector. So that is why it is called as a glue. And finally, we need a bacteria as host. So that means the plasmid, the vector which is cut, the gene which is cut is joined together with the help of glue and then put inside a bacteria, which serves as a host. So this is the diagram which shows the chromosomal DNA, which is the bacterial DNA, and over here is the small circular, mostly circular, extra chromosomal DNA, which is plasmid. And these plasmids, so this is the typical diagram of the plasmid, and this is the typical diagram of the chromosomal DNA. This is a name of a restriction enzyme. You don't have to remember. You have a question? Yes?
So I wanted to ask,
Yes?
what is the use of a plasmid if there are already chromosomes?
Yes. So the plasmids, okay, so chromosomes are there and they contain genes which are essential for the viability, which are essential for the survivability, right? But plasmids are extra, and plasmids are really small. So for example, if I talk about chromosome is like 4.6 million base pairs. What is base pair is ATGC, right? What plasmids, they are very small, 2.6 kilobase, really small. If we put our gene of interest, so let's say I am interested in producing a plant protein, for example, gluten, right, or some other protein. So in such cases, you can put the gene in this plasmid. And this plasmid can be put in any host, for example E. coli, for example Salmonella, for example Rhodococcus, for example Mycobacterium. It can go anywhere. Chromosomes cannot. Why? Because chromosomes have one origin which is specific to the host. Plasmids have origin but can have origins which work in multiple hosts. One advantage. Second advantage is plasmids can have more copies. It can have even 500 copies. Chromosome, 1 copy. So 500 copies versus 1 copy, you can imagine the gene that you have cloned. For example, you can take any, for example, the gene for green fluorescence protein. 1 copy versus 500 copies is a lot. Right? So that is why plasmids are used, because they are used as vehicles. You can overexpress, you can overproduce that protein of your interest, right? Okay. Okay, so here this is the plasmid, and over here this is the chromosomal DNA, and this is the typical name of a restriction enzyme, which is the biological scissor. You don't have to remember the name of the restriction enzyme, just remember that you are using a restriction enzyme. So when you cut the chromosomal DNA with restriction enzyme, the plasmid with also restriction enzyme, they both can be joined with the glue that I was talking about, which is a ligase. So they both, that means the plasmid DNA and this can be ligated with the help of glue, and then you can have multiple combinations where in one of the combination you see that the DNA fragment of your choice is now glued to the plasmid DNA. So for example, the one which is in C is the desired construct, which has the plasmid DNA and part of this DNA put here, correct? And the source of this DNA can be from anywhere. It can be from any eukaryotic system, it can be from any prokaryotic system, it can be from us, right? So far this was about microorganisms in our body, microorganisms in the environment, and I also discussed about how we can engineer these microorganisms for various applications, whether it is production of therapeutics, whether it is production of certain surfactants, etc. So now I thought I will give you a brief outline of what we do in our department. So this is my department of Biochemical Engineering and Biotechnology, and I will be specifically talking about the research related to microbial work. So I will start with Professor Srikrishnan and Professor Zia. So they work in the field of wastewater treatment, and in this case they are using microorganisms for the wastewater treatment, right? And here they are not using one microorganisms, but they are using a consortium of microorganisms. So what is a consortium? Consortium means mixture, right? So when we say pure, that's one, but when it's a consortium, then you can have many types of microorganisms. Some could be culturable, some could be non-culturable, you do not know, right? So they are using microbial consortium for wastewater treatment, and they have carried out these experiments at various levels, lab scale, pilot scale, industrial scale, and they have used for the remediation of even the Barapula drain of Delhi. Next is Professor Shilpi Sharma. She works in the field of plant microbe interaction. So her work mostly has two aspects. One is in the field of plant microbe interactions, where the focus plants are tomato, vigna radiata, etc. and they are developing a consortium, a synthetic consortium. They call it rhizobiome, rhizosphere engineering. And the second aspect is also on fabric microbiome interaction, where they are studying the microbes which are there on the surface of fabrics. So this research is very, very useful, because the fabric which is there on the chairs, for example, where you are sitting, so there can also have microbes. So what should be the fabric of choice if you want to prevent the adherence of the microbes to the fabric? Professor Lucinda, she works in the field of electromicrobiology. So she's trying to uncover the diversity, the various electroactive microorganisms. Finally, she's also trying to develop a biosensor, which is shown over here, for detecting various organisms. This is me. So I work in the field of bacterial genetics as well as environmental biotechnology. So my work mostly revolves around understanding plasmid and chromosome dynamics. I also do a lot of gene regulation, genome engineering. We also work on biodesulfurization, which is removal of sulfur from crude oil. We also do asphaltene biotransformation. So asphaltene are highly complex compounds which you see over here. So in the last panel here, so these are highly complex compounds and they cannot be degraded or biotransformed easily. So in our lab we have come up with a microbial consortium which can biotransform asphaltene. And as a result of biotransformation, surprisingly we found that the asphaltene gets converted into what is called as porous carbon, and it is this porous carbon which can be used in filters as well, for example for water filtration in RO plants. And then we are also doing plastic biodegradation with the help of enzymes. The last part of my work also involves characterization of biosurfactants, which is what we have planned as an activity today. So I will be discussing that. We have Professor Atul Narang also. He works on microbial systems. His main components, the two main areas in which he works is systems biology of gene regulation. The focus is mostly on operons. I hope most of you, I'm sure you would not know what is an operon. So operon is a cluster. Do you know what is an operon? Okay. So operon is when 2-3 genes are present together as a cluster. You know what is a gene, right? So if 3 genes or 4 genes, more than 1 genes are present in a cluster, then it is called as an operon. The expression is driven of, the expression of all 3 genes is driven by one segment of DNA. So that is called as an operon. He's also working in the field of production of therapeutic proteins, bioethanol and certain organic acids. The next one is Professor Ashish Mishra. He does a lot of strain engineering and pathway engineering, and mostly in the field of production of pharmaceuticals, biofuels and fine chemicals. Lastly, Professor K.J. Mukherjee, he works in the field of recombinant protein production, and these recombinant proteins are mostly therapeutics. I hope the students are aware of the word therapeutics that I am using all the time. So therapeutics are compounds which are used for, as the name it says, therapy. So they are used for therapy. So that is why the term is therapeutics. So that's all for the talk, and now I am open to questions. Feel free to ask me any questions that you have. And we also have Professor Shilpi Sharma here. She is also a core microbiologist. So we both of us can take questions. And then while we are doing the activities, if you have questions, ask me. Yes.
Ma'am, you said that plasmid is used, can be used in any of the hosts. Are there any limitations to plasmid hosts?
Yes. So the answer is yes, because when we talk about plasmids, plasmids can be having narrow host range or they can have broad host range. So broad host range plasmids can go in any host, whether it is gram-negative bacteria or whether it is gram-positive bacteria. But narrow host range plasmids will go only in few limited hosts. Yes.
So, ma'am, are there any plasmids that we are deriving from any other bacteria and we can use in the gut microbiomes in the human bodies?
Yes, yes. So in the gut, for example, the strains that are there are lactobacillus, bifidobacterium, etc. So in this, yes, the plasmids can go, and in fact, the whole thing is organized by a club of IIT Delhi. This is a synthetic biology club, and in this, one of the project of this club is to engineer the gut probiotic, and to this we are doing with the help of a plasmid only. Yes.
Ma'am, is there any kind of restrictions or can if we do modifications with plasmid, is there any chance that the microbe rejects the plasmid and actually like
Yes.
a pathogen?
So very nice question, because you are bringing a phenomena which is called as surface exclusion. Surface exclusion is when the plasmid is rejected, right? So this is there only in some cases. For example, coli-1 is excluded from the entry for several hosts. So that is there, but this needs to be tested. Yes.
Ma'am, like in what conditions could this happen?
Say it again.
Ma'am, in what conditions could it happen?
In what conditions? So this is depending on the host. Yes, it is host specific. Any other questions anybody has?
Ma'am?
Raise your, yes. Students, no discussion please. Otherwise we will not be able to start the activity.
Ma'am?
So I would like to have 3 volunteers. Yes, come. First come, first serve. Just 3. At least one boy. Come. Or only 3.
I am also having a question, ma'am. I think so. In first slide, there should be
So we have a method in place by which we can find out who all are talking with their neighbors, and they will not be allowed to perform the experiments. So concentrate on the experiment, right?
We'll discuss. So we need how many? 4 volunteers, right? We have too many. So 1, 2, and 3, 4. Maybe the rest of the students. So maybe students, we can have only 4 volunteers. I am sorry. Rest of the students will do it there. It is not that you will not do it. You will do it there. Please. Mike. Mike. Mike.
Sir, cordless mic.
I will tell their names now. Tell your names and school.
Aditya Pandey, Kendriya Vidyalaya, Sulgaon.
Okay.
Hi, my name is Diana. I'm from boarding school to Sunday Hall. Good morning to respected professor, teachers, and my dear friends. My name is Lalika from IIT Delhi.
So we have four students. So they will be doing the staining. So go there. go there. No discussions, students? No discussions?
Ma'am, I had a question. Ma'am,
मेरा गला बैठ जाएगा.
ma'am, I had a question.
Yes.
Ma'am, once you said that there are certain viruses which are actually beneficial for us and they can infect the bacteria, but then won't be left with viruses in our body, won't they harm it?
Right, so am I audible?
Yes, ma'am.
So the point is that so the viruses that are used, they are not infectious viruses, they are good viruses, with their purpose is only to kill the bacteria.
Then what happens to those viruses after their purpose is
They
served?
will be eliminated with a
Can we request the school coordinators to take care of their groups, please? The school coordinators?
Hello. A1, check.
Okay, so students, everybody got the gloves? Please raise your hands. So I still see many of you haven't got.
Hello, hello. Yellow, yellow.
Gloves. Please wear the gloves. Everybody please wear gloves. Everybody please wear gloves. Show me your hands. Fast. Fast. How many of you have not got gloves? Show me your hands. Student volunteers, please give gloves. Okay, so for those of you who have got gloves, let's get the activity started.
Dear students, let's maintain the decorum, please. We are going to start with the experiments. Please maintain the decorum.
So students, you will be given 2 tubes. You got? How many of you got the tubes? Show me. How many of you got the tubes? Yes, very good. Okay, so the experiment is, तो mic is not working लग रहा है. So the experiment, yes. Students, let's get started, please. Everybody got the gloves? Everybody got the tubes? Very good. So the first activity is biosurfactants. So what are biosurfactants? How many of you know what are surfactants? Surfactants are actually chemical detergents, right?
Preeti, wait for the crowd to maintain the silence. Wait for the students to maintain silence. They're not interested in watching the interesting video and the other activities that we have planned. Are we all now looking at the screens, students? We have something special.
Do you want me to start the second activity? Then don't make noise, please. Can I request? Can I request? No noise, please. So now we are giving you another set of 2 tubes. And this is again the same experiment that we did just now, where you will be mixing the solutions of both the tubes in one. Tube number 1, right? These are the new tubes. Tube number 1 actually contains a bacterial cell suspension, and tube number 2 actually contains the lysis solution. And I'm going to show you that how genomic DNA comes out. So, how many of you got the second set of tubes? Please raise your hands. In the middle row, I think students haven't received. Everybody got the 2 tubes, students? Everybody got the 2 tubes? Please raise your hands. How many of you got the second set of 2 tubes? Okay, so let's get started with the second activity. So in the second activity, again take out the 2 tubes. Take out the 2 tubes. Mix the solution. But this time, you need to be gentle. Don't do vigorous shaking. You need to be gentle. Do slowly. Take out the 2 tubes. Mix the 2 solutions. You need to be gentle. Do not do vigorous shaking. Mix it gently, and what you will see is
the solution getting clearer, and the solution will become viscous. And if you open the lid, you will see the strands. Open the lid. Open the lid and see the strands. Do you see some strands? Yes. So that is actually genomic DNA. That is actually genomic DNA that you have isolated. Right? So I repeat, in this activity, we are actually trying to do genomic DNA isolation. And in genomic DNA isolation, we are mixing the content of one tube with the other tube. Tube number 1 contains cells. Tube number 2 contains lysis solution. You will see that as a result of lysis, the solution becomes clearer, and then you will see strands of genomic DNA. Are you able to see? Tell me yes or no? No? Okay, open the lid. Open the lid. 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 Yes, now you can see. Open the lid. Tilt it and open. It's viscous. So you will see the clearance zones. Right? So while so continue with your second activity, and let's get started with the third activity.
अच्छा, लेफ्ट साइड वाले मुझे बताएंगे कि उनको एक्टिविटी करनी है कि नहीं, फाइनल वाली. लेफ्ट साइड 1 2 3 यस. इतना धीरे धीरे यार. बीच वाले 1 2 3. राइट साइड ना ना ना. फर्स्ट उधर फिर से एक बार चांस मिलेगा. 1 2 3 यस. बीच वाले राइट साइड. वंडरफुल वंडरफुल ग्रेट. तो प्रीति मैम, आप दिखाएंगे एक्टिविटी क्या है?
Students, so the last activity for today is view your microbe. View your microbe. So we had 4 volunteers. And we have a sample. What you see is a sample of yeast. The slides are prepared by a student, one among yours. And I will now call upon the stage the student who has actually prepared the slide. So who has prepared the slide?
Hi everyone, my name is Anushka and currently pursuing BTech from Kalam. So here I found a new experience with ma'am. Thank you for giving us this opportunity. So I have tried one experiment which comes in activity, and this is where I have done one experiment of wet one, where I have kept one slide which I need to first clean that with the tissue paper, and then I have I don't know the name. Yes, stain with that, and after that we have cleaned it. Then we have put on that with the pipette, I guess pipette, yes, with pipette, and then we have departed that from on the slide, and then we have covered it with, again we need to clean with that, what is that small one? I don't know that. Yes, a small one, a piece of type of paper which is very slightly very light, and then I have done this. Thank you.
So that was, what's name?
Anushka.
That was Anushka. So Anushka has prepared a beautiful slide. Thank you, Anushka. So now we will call the next volunteer. So is the next volunteer, the next student volunteer? What did you do?
Hi everyone, so they made me take the culture and I had to take a sample from the rod. I had to rub it upon the slide and I had to let it set for a while. We heated it and then we took it and we just, I don't know how to explain, we just put it across the heat, the flame, right. And then we let it set. After that, we took the dye, we put it on, then we washed it with water, and we let it dry, and then we took it here, and I mean, you can see the cells.
So thank you. What was your name? Shaina. So thank you. Raina. Thank you, Raina. So we will now call the next volunteer, the next student volunteer.
Ma'am,
I'll have to hand over the slide.
Okay, that will be much easier. Thank you.
So this one.
So this would be red, right? No, red. Pinkish, correct.
yes, ready.
You go and explain. Bolo.
Okay, so this is an experiment of gram staining. So first we took two bacteria. We did not know which one, but one is gram positive and one is gram negative. Then we took a slide. We first cleaned it with a tissue paper and ethanol to remove any other germs that were present on it from earlier. After that, okay, gloves is a necessary part of this. We have to wear gloves as to prevent any microbes from our hand or to prevent our hands from staining as well. Then after that, we took a pipette and then we set it to 5 first. On the pipette, we set it to 5. Then we took some portion of the bacteria from the tube. Mine was E. coli, so I took some E. coli and then I pumped it on the slide. Then using a loop, we smeared it properly so that it is easier to view. After that, we heated it. We heated it so that it fixes on the slide when we wash it with water after the next step, in the next step. After washing it with water, we put some amount of crystal violet stain on it. Crystal violet stain is the gram stain here. After that, we washed it again. Then we put some of the iodine to fix the stain there. After that, we washed iodine and put safranin. See, the gram-negative bacteria have a thin layer of peptidoglycan and the gram-positive ones have a thicker layer of peptidoglycan. The thicker layer of peptidoglycan allows the gram-positive bacteria to retain the gram stain, and the thinner layers does not let it retain the stain, hence it gets washed away when we decolorize it. And hence when we put safranin again, we can see the pinkish color, but in the gram-positive bacteria, the crystal violet color remains because it does not get decolorized. That is what we are able to see there. Thank you.
Bhavya. So very nice, Bhavya. Last volunteer. So this is the last slide on view your microbes.
Hello.
मैंने अभी यहाँ पे डायोइक से मेरे को बैक्टीरिया दिया गया था, तो मैंने उस पर उसको साफ़ किया। पहले प्लेट जो होता है ग्लास प्लेट, उसको साफ़ किया, फिर उसमें बैक्टीरिया डाला, और फिर जो बैक्टीरिया डाला उसको हमने जो हीटर होता है उसके हीट किया, ताकि जो बैक्टीरिया होते हैं वो उसमें चिपक जाए। और उसके बाद हम लोग उसको आराम से अलग अलग चीज़ों से साफ़ करते हैं, फिर उसको स्ट्रेन करते हैं। आप देख सकते हैं यहाँ पे बैक्टीरिया दिख रहे होंगे। अगर हम लोग साफ़ नहीं करते तो बैक्टीरिया नहीं दिखते। और ये कलर ब्लू है।
Okay, so thank you everyone. So now my PhD student volunteers will be collecting the gloves and the packets from you. So please deposit all the gloves and the packets. They will be collected back. I will show you a small video while the students are collecting the packets. Can I? Okay, so this is the microscope that was used. This is called as otoscope, and the name of the company is IIT Innovations. The founder is Professor Ravi Krishnan Elangovan, and this is our own in-house microscope which has projection facility. So this is the one which was used for projecting it. As part of the continuing the celebrations of the International Microorganisms Day, we organized 2 hackathons. And I will show you the video. Sound. So 16 states. So the hackathon of microbe innovation was conducted as part of International Microorganisms Day. The participants were school students as well as college students. And now I'm going to announce the winners of the hackathon. We had participants from all across India and even from UAE, Qatar. So we have participants not only from India, but from outside. And now we would like to announce the names of the winners. So the winners are, for Design Your Microbes, this is a school level competition. And the first prize goes to DPS Monarch International School, Qatar. The second prize goes to Ankush Rai, Adifa Farzeen, Sara Shamim, Ayushi Kapoor from school Sarvodaya Kanya Vidyalaya, Noornagar, Convent of Jesus as Mary, and Arvachin Bharti Bhavan Senior Secondary School. The third prize goes to Anurag Kumar from DBRA Sector 6 Dwarka. So So the next hackathon is at the college level. And at the college level, the hackathon is Microbe Innovation Ideathon. And the first prize goes to Guru Govind Singh Indraprasth University, Delhi. The team has Dhwani Goyal, Anushka Raghuvanshi, and Bharat Bharadwaj. The second prize goes to Maharaja Agrasen Institute of Technology, Delhi. And the third prize goes to Atharva Narale, Lucky Agarwal, Sandesh Jadhav from JSPM's Bhivrabhai Sawant Institute of Technology and Research, Pune. So I would like to finally close my part. So I would like to thank Professor Shilpi Sharma. Of course, my students, Nidhi Patil, Ashimita Bagchi, Diksha Jain, Aditi Keshav, Purvika, Archana, Gursimar Singh, Anish Razu, Renu Mazu, Monica Avasthi, Sai Ganesh Nair, they all are here, and without them, this whole program would never have been seen the day of light. The support team at DBEB, that's my department and outreach office, Shivani, Ratnabali Ghosh and Mohammad Sinad were on the microscope, the otoscope that you saw. Dr. Sumit Kapoor also helped, Ankita, Mayuri Rajput, Avnish Kumar, Sanjay and Ashish. Thanks to all of them. I would end this by playing a video, and I would request all of you to see this video. It's a long video. There are 4 volumes. I will play the trailer and then I will hand it over to Shivani.
What if your life, all life, depended for its very existence on creatures that dwarf us in numbers, yet few of us have ever seen? It does. And what if one day life on Earth began to disappear, and we didn't know why, because we didn't understand these creatures' power to control the environment? It could happen. But perhaps it won't, if we learn to understand that we are not the true masters of life on Earth. Microbes are.
Strangers, Keepers of the Biosphere, very beautiful volume. I have given the link of the video as well in the booklet that I have shared with you. It's a must watch.
Thank you, ma'am. Students, the list of the winners will be hosted on our academic outreach website, which is prominently mentioned in the details of our official WhatsApp channel and also in the live streaming video on the YouTube. You can find the same in the description link. With that, thank you so much, Professor, for your valuable insights. Your words have truly enriched today's session. With that, we are now open for question-answer session. We invite our audience to share your questions, thoughts, and reflections. This is your chance to engage directly. So please feel free to raise your hands. We'll come to you for your questions. Any questions, students? No questions? Okay.
I've heard some things about CRISPR-Cas9. What exactly is that? I've heard some things about CRISPR-Cas9. What
Yes.
exactly is that?
Oh, CRISPR-Cas9, it will require a longer discussion, but CRISPR-Cas9 is actually bacterial immune system. So it was found that there are certain sequences. Maybe let me tell you in a simple way. So bacteria accumulates these sequences every time when a virus infects a bacteria. Right? So bacteria acquires an immunity. This is called as adaptive immunity. Maybe I can tell you offline. I can draw it and explain you.
Okay, ma'am.
Good morning, ma'am. My
Good
name
morning.
is Mitali. Basically, I want to ask that bacteria is actually good for our body. Like it's a blessing in disguise, right?
Yes.
So, ma'am, how it's like infecting of bacteria is beneficial? Like when virus infect bacteria, you said it's beneficial. So how it is beneficial? Like I didn't understand.
Yes, so this is a new concept which is coming up, which is called as phage therapy. Phage therapy. This is a new concept which is coming up. This is like a substitute of chemical antibiotics because people realized that these chemical-based antibiotics are doing more harm than use. So that is why phage therapy has been suggested as an alternative mechanism. And the good part is that when we talk about phages, we can have very, very targeted killing. So for example, let us talk about the gut microbiota. So in the gut microbiota, you have beneficial microorganisms as well. But then if you selectively want to target, let's say Shigella, right? So then you do not want, if you are using an antibiotic, it can have broad range, it will kill all gram negatives, all gram positives. It cannot specifically target Shigella. On the other hand, if you are using phage therapy, then we can have specific phages which will not target others, but specifically infect Shigella and kill it. That is why.
Thank you, ma'am.
Good morning, ma'am. Is the gut ma'am? Good morning.
Yes, good morning.
Is the gut microbe different in every person, like a fingerprint?
Yes, so that as I said, I showed in one of the slides. So about 160 microorganisms, like 160 strains are conserved in all of us. But then there are many, many which are different. And that is like a signature for every individual. But then it is dependent on the food, on the dietary habits, on the environment and other things. So when we are born, the microbiota that is there, that is like conserved for 3 years, and after 3 years, as you grow up and as you experience different environments, different food habits, it kind of changes. But then again, when you become old, like 55 or 60, the moment we reach 60 years of age, again we do not see much change in the microbiota.
Thank you, ma'am. Thank you, Professor, for your grace, for bringing passion to this place. Your words will echo strong and true. We are grateful for the gift of you. It's truly an honor and a privilege to stand here today to express our deepest gratitude to Professor Preeti for taking time out of her busy schedule to be with us and to share her valuable insight with our students. Your lecture today was not just informative, but truly inspiring. Your words of wisdom has enlightened every one of us. As a small token of our appreciation and respect, we would now like to present you with a memento. This is not just a symbol of today's event, but a heartfelt thanks to you for your gracious presence. May I now request Professor Shilpi Sharma, the Associate Dean of Academic Outreach and New Initiative Office at IIT Delhi, to do the honors. Ma'am, please. Thank you, ma'am. Can we now capture the moment with a group photograph? Ma'am, please. We'll take a group photograph. Thank you, dear students. Thank you, ma'am. With that, I hereby wish to announce our next Sci-Tech Spins Lecture Series, which is scheduled on 11th October forenoon by Professor Jayadeva from the Electrical Engineering Department on the topic Ant Trails and Viral Trends, the link between swarm intelligence and social trends. Please have a look at it. I request the school coordinators to send us the nominations. We will soon upload the information at our academic outreach webpage. With that, students, we have shared a link to you, which is a QR code of feedback. We would request you to send your feedbacks ASAP. A big thank you loud and clear. For your support, we truly cheer. With smiles and joy, we say today, thanks a lot in every way. With that, I now take this opportunity to express our gratitude and heartfelt thanks to all the students and the coordinators. With this, I, Shivani Kush, take your leave from the stage and leave you all with a beautiful quote. Dreams take flight when courage leads. Hope is planted through humble seeds. With faith and effort, the path shines bright. Every dawn brings a brand new light. Brand new light, brand new light. Thank you so much. Dear students, we wish to show you a QR code, and I'll take 1 minute of yours scanning that QR code, and the students who are not carrying, I'll request the coordinators to do the needful so that the feedbacks can be shared to us ASAP. Thank you. Please scan it out and give us your feedback. Your feedback is very valuable to us.
The QR code is also there in the booklet that I have given you. I would request Professor Preeti to be on the dais with the volunteers for a group photograph. I'll request all the volunteers to be on the dais for a group photograph. Coordinators are requested to send us the nominations of the students for the next Sci-Tech event, which is on 11th October in Seminar Hall. This is second Saturday. The timings are 10:30 till 12 p.m. With that, students, I'll take your leave. Thank you so much for listening to me. The exit gate, the refreshments are lined up there. Before you leave the premises, do enjoy the refreshments. Thank you so much.